Hello, General Biology, and we're still continuing the screencast series on Chapter 2, and this is Section 2.3, and this is the third of the screencast for 2.3. And we were talking about lipids when we left off, and lipids are complex macromolecules. They're giant molecules. We talked about them being made up of glycerol and fatty acids. And there's different types of fats that we're going to be mentioning in just a minute. We said that they are insoluble. They cannot be broken down by water, and we can see why based on the bonds. And we're going to look at those bonds in a little bit more because we have two types of fats that we're going to discuss, and they are saturated fats and unsaturated fats. And saturated fats, we're going to find these as being solids during room temperature. So that's going to be your butter, your lard, okay? They're solid. And unsaturated fats are going to be liquid at room temperature. So those are going to be your oils, your corn oil, your vegetable oil, your canola oil, your peanut oil, anything that's an oil. Okay, so when we talk about saturated versus unsaturated, saturated means that every carbon is holding two hydrogens in the way that they bond. Where unsaturated means there's some carbons. So as I was saying, the dog barked, so excuse me. When we have unsaturated fats, then that means that each carbon is not holding two hydrogens. So let's take a look at our fatty acids. And we're seeing all the way up to here that our carbons are holding two hydrogens, except for these two. So when I look at that, and I'm trying to determine, is that going to be a saturated fat or an unsaturated fat? That to me is going to be unsaturated because the carbons are not holding all of the hydrogens that they can. So when we have a lipid molecule like this, we could have, let's say, two of these fatty acid chains being saturated and only one of them being unsaturated. We could have two of them being unsaturated and one being saturated. So we can get a combination of these saturated and unsaturated fatty acid chains that are in there that would make the molecule have the tendency to be a liquid or a solid at room temperature. I hope that makes sense to everybody. If it doesn't, then ask me and we'll go over this in class. And don't be afraid to ask if you're not sure. Because this is new for everybody. So if each carbon atom in a lipid's fatty acid chain is joined to another carbon atom by a single bond, that lipid, as I said, is said to be saturated. Okay? If there's at least one carbon-carbon double bond that we saw in the fatty acid, then it's said to be unsaturated. And that's because it can't hold the hydrogens, like I had mentioned. So think of it this way, if you're saturated, you're holding everything you possibly can. Think of those heavy book bags that you have. Oh my gosh. And if you have every single book in there, you're saturated. You're holding everything you possibly can in that book bag. But if you're unsaturated, then there's extra room. You don't have your book bag totally filled. Okay, so that's a way to remember it. Lipids whose fatty acids contain more than one double bond, so if we have more than one, not illustrated in our picture there, it's said to be polyunsaturated. And when you're thinking in terms of lipids and what's a good lipid and what's a bad lipid, you want unsaturated or polyunsaturated fats because they're already liquids at room temperature and your body doesn't have to turn them into liquids to break them down. So liquids that contain unsaturated fats, as I gave examples, are olive oil. They tend to be liquid at the room temperature. And we can look at the different types of fats that there are. There's stearic acid, oleic acid, linoleic acid, and linoleic acid. Um, and we can take a look at the number of double bonds that they have. So when we look at stearic acid, one of our fatty acids that make up a lipid, and we see that it doesn't have any double bonds, its melting point is high. It's huge. It's 69.6 degrees Celsius. So, okay, if you're not really comfortable with Celsius, 23 degrees Celsius is room temperature, so 72 degrees. 37 degrees Celsius is body temperature, 98.6, okay? So when you're talking about 69.6 degrees, that's hot before it melts. We can look at oleic acid. It has one double bond, and its melting point is 14 degrees Celsius. So 0 degrees Celsius is freezing. So that's above freezing. It's not as quite as warm as room temperature is. We'll skip down to linoleic acid, and that has 3 bonds, and the melting point, look at that, minus 11. It can melt at below freezing. Wow. Okay, so that is some of the chemical properties of these lipids. And as I said, a lipid can contain any combination of these fatty acids, making it unsaturated or saturated. The next compound that we're going to talk about are essential to living things, and they're called nucleic acids. This is so important that we're going to spend later chapters talking about them. We'll talk about them again in chapter 7, and then it has its own chapter devoted to it. I believe that's chapter 10 and even 12, and we'll talk about it when we get into our genetics unit. So this is a very important macromolecule. We're only going to touch lightly on it here, but just make a mental note that this guy's huge. And you know, a little play on words, it's a macromolecule. It's a huge macromolecule when we come to see it. So nucleic acids are important for transmitting our hereditary information or our genetic information. It's the reason why you resemble your parents. It's the reason why that you have a nose centered on your face in between those two cheeks rather than the two cheeks that you have that you sit on. And thank God for that. It's the reason why you have blue eyes or brown eyes or hazel eyes, the color of your hair, how tall you are, whether you're going to have dimples. So all of that is your hereditary information, and it's contained in these nucleic acids. So everything about you, the whole functioning of you, happens because of these nucleic acids. Nucleic acids are macromolecules, and they contain hydrogen and oxygen. We're going to see the addition of nitrogen and phosphorus to our chemical formulas for this. They are polymers. They are assembled by tiny monomers that come together, and they make up compounds called nucleotides. And we're going to talk about those nucleotides more. We'll watch this video in class if you can't click on it. I know sometimes they don't work. Okay, so let's talk about these nucleic acids. As I said, they're made up of nucleotides, and the nucleotides, before I even do that, let's go back a minute. And I just want to say there's 2 nucleic acids, and one that you've heard, and I'm sure you've heard of both of them, deoxyribonucleic acid. Otherwise known as DNA, and this is the master molecule. And then RNA, which is ribonucleic acid. and this is a helper. And it's going to help in a lot of ways. Deoxy means without oxygen. Ribo is a sugar, ribose sugar, and it's a nucleic acid. RNA is ribonucleic, meaning it has ribose. Okay, so those are the two molecules that we're going to look at as far as nucleic acids are concerned. So as I started to say, they are made up of nucleotides that actually consist of 3 parts. A 5-carbon sugar, something called a phosphate group, and we're going to talk about those phosphate groups often throughout our lessons this year, and then something called a nitrogenous base. And a nitrogenous base is just a nitrogen-containing base. Remember, we talked about pH, so it kind of gives you an idea that it's going to have hydroxides in it more than it's going to have hydrogen ions. So our 5-carbon sugar, we're going to represent in this diagram by a circle. And that is a pentose sugar, or a 5-carbon sugar. Oops, I'm sorry. That's not it. This is it. I'm asleep this morning. So it's also going to be known as a pentose sugar. Pentose means 5, and we use a pentagon to show the 5 carbons. And we have 2 kinds of pentose sugar. We have deoxyribose, and we have ribose. So that shape could be either one of those. A phosphate group. Now this is the phosphate group. Notice it says PO4, and that's because it's phosphorus and oxygen. So it's a phosphate with 4 oxygen that are joined together to make up that phosphate group. And then the nitrogenous base, a nitrogen-containing base. And there's 4 nitrogen-containing bases. There's adenine and guanine, and there's thymine and cytosine. Adenine and guanine are double-ringed. Monomers, okay? So this could be either adenine or guanine. Thymine and cytosine are single ring in their chemical structure. And when we do bonding, adenine will always chemically bond with thymine, and guanine will always chemically bond with cytosine. One of the things that it's important to notice is nucleotides contain adenosine triphosphate, or ATP. And ATP, adenosine, would be adenine. The syn part would be the ribose. And the triphosphate would mean that they have 3 of those phosphate groups. And that's important in actually chemical energy. And we're going to talk about that later on. But for now, we're going to look at. So individual nucleotides can be joined by covalent bonds, and when they do, they form a polynucleotide. Again, poly meaning many nucleotides, or nucleic acid. And we already know that there's 2 kinds of nucleic acids. And I just want to write this down. This is a nucleotide. Okay? Nucleotide. Nitrogen base, one of our nitrogen bases, a double ring in this case, a sugar, and a phosphate group that's present. Okay, we're going to stop here, and we'll get more into the structure of DNA in the next screencast.